All right, so I'm going to try to talk loud. I don't have my microphone. I lost it. So I'm going to go through and do a review for material that's on the second exam in our course, which focuses on membranes. So we're covering membrane structure, then movement across a membrane, diffusion, and osmosis. And then we're looking at facilitated diffusion, active transport, bulk phase, cell adhesion, cell communication. So I'm going to try to go through all of those right now in this review video, and I'm going to try to do it in just like 15 minutes. So this is like all the lectures of the whole thing, which are several hours long, condensed into just a real quick little summary of everything. So I'm going to be going fairly fast. If you're missing things, and I'm not going to go over every single detail, you have to go back to the actual lectures on those specific topics to get the full information. Okay, so talk about membrane structure first. Okay, membranes, and this is for membranes for eukaryotic cells, which is the focus of our course, which include animals, plants, fungi, protists. But for the prokaryotes, bacteria and archaea, the bacteria have very similar structures. The archaea also similar, but modifications. So the this structure here that we're drawing, or I'm drawing, as the phospholipid. So that's going to be our phospholipid. And you need to know the structure, the full structure, the chemical detail structure. So if you're asked to draw a full membrane and with all the phospholipids in it, you don't have to draw all the glycerol molecules and individual carbons and hydrogens of a fatty acid for every single one. You're drawing this little representation of the polar head and the nonpolar tails. Hopefully after the first exam, you are pretty well aware of the difference between polar and nonpolar, what causes molecules to be polar or nonpolar based on the types of sharing of electrons between the elements of that particular molecule. And what we have for the polar part, there's a glycerol, I'm not going to draw every carbon and everything, there's a glycerol molecule, which is a 3-carbon molecule. It has 3 oxygens sticking off it like this. To these particular oxygens here, that are coming off the bottom, that's where we're going to get 2 fatty acids. And again, I'm not going to draw all the details for time. They could be saturated fatty acids, and in which case there are carbons and hydrogens. That's it. There's nothing, and it's all single bonds between all the carbons, so they're perfectly straight. Or there could be carbon-carbon double bonds, where that means there are fewer hydrogens, so they're unsaturated, and that would cause them to bend like this, and you could have multiple bends, so they can be bending back and forth like this, could be polyunsaturated. That's going to be our nonpolar tail part. Our polar part is going to consist of the glycerol, phosphate functional group, which has oxygens attached to it, and then also a polar molecule that's attached to that. So there's this sort of a big polar area and a big nonpolar area, and that's our phospholipid. That's the basis of all the membrane structures. However, when cells are analyzed for their membrane composition, originally there was like 50-50 for some of the original cells, like protein lipids. So people didn't know if it was mostly a protein structure was the membrane and the lipids just filled in, and how it was all put together. Cells range in the composition to 80% protein and only 20% phospholipids, or vice versa. It could be mostly phospholipid and less protein. Different membranes have different compositions, but most all membranes have some type of them proteins associated with them. Membrane proteins may go all the way across the membrane like this. So we're going to go over here. This one we're going to talk about. That is going to be, for us, I'm calling it a transmembrane protein. Some people call it an integral protein. A transmembrane, it spans the membrane. It goes all the way across from one side to the other. It is present in the cytoplasmic side. And it also has a presence in the extracellular side. And there's usually an alpha helical region that is then embedded in the area of the fatty acid tails, typically the R groups for those amino acids, or nonpolar R groups that are sticking off of those particular amino acids. But there's a lot of variation to that, but that's kind of what happens. So you have sites on each side. that is our, mention there, different colors I could label. That's our transmembrane protein. Versus this one here. This protein is only present on one side of the membrane. We could have ones on the other side, as well. I just drew it on this particular side. It's not exclusive to the cytoplasmic side, but that's a peripheral protein. And so peripheral proteins are embedded in the membrane, but they don't go all the way through. They don't go all the way to one side. So they may be only on the outside and not span all the way to the inside, or they may be only on the inside. In our class, mostly we're going to be talking about peripheral proteins that are on the cytoplasmic side that have different roles in some of these other types of cell function. All right, so we have phospholipids, and then we have proteins, and the proteins we're just generically categorizing as either proteins that go all the way across or attached to one side, peripheral or transmembrane. There's also some other things. Okay, so we have in between, and I'm not going to draw the full detailed structure, just do something like this here. This is representing and I'll do maybe some other ones here, I'll do a couple here. This is supposed to represent a cholesterol molecule. So we don't see the cholesterol molecules in bacteria membranes, but we do in all the other organisms. And the cholesterol molecules have a variety of different jobs. One of the main jobs they have is maintaining fluidity of the membrane, how flexible it is, as a counter to changes in temperature. So temperature will change fluidity, cholesterol will change fluidity, the fatty acid tails will affect fluidity. So essentially it's how flexible the membrane is. The membrane is not like a hard wall. Okay, the membrane, there are no bonds really between these, right? The fatty acid tails being non-polar means no interaction with other things. So they don't like each other, they're not bonding to each other, there's nothing really going on. It's a non-polar association. Okay, so that means there could be movement. These phospholipids can move around, and there can be movement of the membrane itself. So the cells can change shape. You can kind of distort the cell shape. It doesn't just break or shatter because it has flexibility to it. But it has to have integrity to it as well, so it doesn't just fall apart. You can't just kind of rip things apart. So there's other things that we'll talk about, that how the cells bind together with this adhesion part that builds the tissues, which isn't really our course, but it kind of leads in from our course into other courses. And we'll get into it just a tiny bit here. So cholesterol molecule, if temperatures get really high, the cholesterol molecules are kind of like grouping phospholipids together from being able to move too far apart. And as temperatures get colder and the phospholipids are moving closer together and they're not moving so much, the individual cholesterol molecules kind of create little pivot points and spacers, so it allows a little bit of flexibility. So it doesn't make them completely tolerant and unaffected by temperature. It just makes it so that as the temperature increases, the integrity is kept down. The fluidity is kept down from becoming too fluid and falling apart because of cholesterol. And as it becomes very cold, membranes can eventually freeze solid, but cholesterol can change the temperature, making it so that it has, not the temperature itself, the temperature at which it will freeze solid. So they can be a buffer for those temperatures. So the cholesterol is there. The phospholipids are there. And then we also have carbohydrates. So that's what this little thing looks like, a lightning bolt, kind of fancy one, sticking off here. So carbohydrates could be attached to the, I'm usually drawing a branch kind of like this, to the phospholipids, or they could be attached to the proteins. So if they're attached to the phospholipids, they're a glycolipid. If they're attached to the protein, they're a glycoprotein. Okay, so again, for space, I'm going to have to erase a lot of this, so I'm not going to keep drawing everything. But so glycoproteins and glycolipids are essentially just sugar groups attached to the surface. Now the sugar groups can be unique, what sugar they are, how they branch, all these different sorts of characteristics to become like our blood typing, our ABO blood system, O doesn't have any of these. The sugars on the surface of the red blood cells, but A have a unique group of sugars. B has a unique group of sugars. People who are AB have both of those groups of sugars. So it's a glycoprotein that's responsible for it, but what's unique about them are the carbohydrates for the blood typing groups. So kind of keep that in mind. So we have making up the membrane structure: sugars, carbohydrates, proteins, lipids. So we don't have nucleic acids here, but some of the major groups of biological molecules that we covered in the first exam are all present here in the membrane structure, right? And we have proteins that are transmembrane all the way through, a peripheral on one side. Now the membrane creates a barrier, we said, because this area is really nonpolar and it doesn't like things that are polar or things that are charged, okay? And so put water, put a couple different things here. All right? And I'm also going to put this. This is, that's oxygen, okay, molecular oxygen. So now molecules are going to move from any area that they're in to other areas based on their concentration. So they're going to, so let's say we had just a chamber, for example, like this, and I put some water in the chamber, like this. And then I added a little barrier between two sides. Equal amounts of water on both sides. But if more water was added to this side and the water level rose up, you can see what's going to want to happen. The water's going to want to move to that side. It can't because there's a barrier blocking it. But if it had a way to do so, it would. So one, this is storing energy. There's energy on the high concentration side. High concentration, high energy. And I want you to really focus on that and keep it in mind because it is important for the content of this exam as a concept. It's going to be extremely important for your third exam as a concept because this is how cells are going to convert molecules stored at high concentrations from one type of energy to another to the usable energy of ATP. So, or essentially, putting it a different way, how do cells make ATP? Where does the energy come from? Most all that energy comes from the storing of molecules on one side of a membrane in a concentration gradient and then releasing that and then allowing them to flow through to the other side. And as they flow through, they'll release that energy. And that energy can then be used to do some type of work in the cell. So cells will be doing this using differences in concentration of ions and certain molecules across the membrane. Now, how are those molecules and ions going to get across the membrane like this? Well, one way to get across is, directly between the phospholipids. But we have to ask for certain molecules, how likely is that to occur or not? These sodium ions are positively charged. Do they interact well with these tails? No. They don't like the tails don't like them. This area here, this kind of pinkish nonpolar area, is a barrier for anything that's charged. And so typically, no, they won't. Will they go through? The answer is no, typically not. They're not going to be able to get through the membrane. So even if there's a lot of sodium on one side and it would like to get in to the other side, so let's say instead of just water, this is sort of a concentration of sodium ions kind of built up on one side versus the other, they would like to flow through, but there is a barrier blocking them so they cannot get through to the other side. Here's an oxygen molecule, so O2. Right, that's what we have here. Now, oxygen, remember, is very electronegative, which throws people off, thinking that, it's going to be polar. But if an oxygen is attached to another oxygen, they're equally electronegative, so they share electrons equally, so there is no charge. So it's not polar. It's a non-polar. So, does it have any problem with the tails? No, there's no charge here at all. So gases, right, like nitrogen and oxygen can easily then pass across membranes from one side to the other purely based on concentration. So high concentration on one side, low on the other, they'll just move across the membrane. They don't have to be helped or assisted typically in any way because they don't carry the charge. But charged things typically are set up as a barrier. Bigger things are set up. So if we have a sugar molecule like glucose, it's not just 6 carbons and 12 hydrogens and 6 oxygens, you can start to say, oh wow, hydrogens and oxygens here, we got a lot of probably polar bonds, right, in a carbohydrate. In addition to that, it's going to be bigger. It's 6 carbons long. It's not going to fit through between the little phospholipids, and it's also not going to like that environment because of the polarity. So it's not coming through, and not directly through. Okay, so this is where then the proteins are going to come in. So I'm going to do a little bit of erasing of some of these things. Okay, just so we can see what's going on. Just erase the cholesterol, just so I can write some other labels and things too. Okay, so how do we get these things across the membrane? They're going to come through proteins. So one of the jobs of transmembrane proteins, and keep this in mind, what is your second exam all about? Well, it's about membranes, but it starts off with the structure of the membrane, but it's really going to be about the job of membrane proteins. Membrane proteins are going to be involved in facilitated diffusion. Some of them are going to change shape, as what we're going to talk about as carriers. Some of them are not going to change shape. They just form little tubes, channels through the membrane, and they're going to allow diffusion. That'll be facilitated diffusion. There's going to be some that can move things the opposite way. So take the sodium ion and actually push it against the gradient. So I'm sort of like pushing this way. So how would you get the water to flow from this side to that side? That's not going to want to happen, right? But if you use energy, you can pump it. You can push it in that direction. And so some proteins can then couple reactions that involve use of energy and a whole variety of different types of energies. That's another part of active transport, a big thing. I'm not going to be able to get into all of it, but is where does the energy come from for the active transport? There is no one answer for that. It may come directly from ATP. It may come from a bound phosphate, which was indirectly from an ATP previously, but now it's been broken off and attached to the molecule, so it's a second bond-breaking reaction that releases energy. It could come from concentration gradients. It could come from electrons, like in photosynthesis, so or electron transport chain. So all these different ways that energy can be transferred to a protein to allow it to do this type of work. But the bottom line is we're talking about the protein, which is a specific shape that's going to change shape, and it's going to move something across the membrane, and it's either going to move it with its gradient, so from high concentration to low concentration, and if that's the case, that's going to be diffusion. If it moves it the other way, like this, if it's the sodium moving from low to high concentration, that's going to be active transport. And that's going to require some energy, right, in order to do so. So before we get into all the details of that, we still have water sitting out here. And so water is something extra to consider, right? We have a molecule, we have an oxygen atom and then 2 hydrogen atoms, but right, it's not linear like this. They're angled so that there are sides. So the oxygen side is partially negative in charge. The hydrogen sides are more positive in charge. I don't know if it's positive or negative. Oxygen is negative, hydrogen side is positive. So we kind of have 2 sides to this molecule. It is a polar molecule. Water is polar because of the unequal sharing of those electrons and because of the shape of it as well. See, I'll bring this in really quick here. Carbon dioxide, CO2. Technically, the bond between the carbon and oxygen is polar, as well. But what happens here is we get like this. So it's linear. It's a straight molecule like this. And so while this would tend to be partially positive because of those bonds, these tend to be partially negative, that positive charge is sort of trapped to the interior of that molecule. It doesn't have the same sort of effect as the water molecule does. So it behaves in a little bit more of like a slightly partially charged or even potentially canceling each other out, non-polar type of molecule. So carbon dioxide as a gas will behave like the other gases, as well, and just kind of diffuse across membranes. But water won't necessarily just move across as easily. It depends on a whole lot of things. So can it diffuse directly across the membrane? So can water just move right in between these? Yes, that's what we call osmosis. Okay, osmosis, water is just moving right across the membrane. How easy is it for water to move? It depends on many things. It depends on the packing of the tails. If they're all straight tails, the membrane's packed a lot tighter, so it's a little bit more difficult to happen. Whereas if they're like this and they're bent, there's a lot more space, so it's a little more fluid, and water can have an easier time getting through. It has to do with things like membrane potential and charges and how well it can get across. It has to do with temperature, really cold temperatures, again, tight packing versus warmer temperatures, and a variety of other sorts of things. So yes, water will move across, but cells can also regulate it, and sometimes they'll use proteins to actually push water in one direction or another, but most of the time they're pushing ions to offset the movement of water, because water is going to move pretty much freely on its own one way or the other. How can it do it if it's polar? Because I said these things can't. Well, one is, this is a true charge because it's an ion. These are partial charges, which means they're in flux. They're constantly, so this is negative sometimes, it's neutral sometimes, and positive sometimes. Most of the time it's negative. So if it was timed, how long is this one negative? Most of the time, but not all the time, because it's a partial charge sort of effect going on there. So there's this flux happening with them, and that affects it as well. But water can move across. There's also pressure from water going from one side to the other, kind of like from this example. So water will be moving across regardless. It doesn't necessarily have to have the help of a protein. But for all these others, they have to have the help of a protein. So some of them will be able to move through a channel, and the channels can not just be a hole that anything can move through, but they can have some regulation. So a channel like this, let's say for example, if it had a lot of positive charges on the R groups inside it, would the sodium want to go through? Well, from high to low, yes, it would want to move through, but would it be able to? Once it gets in this environment, the positive charge is repelling it, essentially pushing it out. So no, it won't be able to get through. Even though there's a hole there, it could fit through the hole, it's going to go from high to low, it doesn't like that. But if this one has more polar charges and some negative charges, then it can flow through from one side to the other. So ion channels can be partially selective. In addition, they're sized. So if this was really large, it wouldn't fit through the opening. So anything isn't going to just be able to go through the ion channel. Ion channels can be gated, meaning that they can have peripheral proteins attached to the transmembrane protein that act kind of like a little plug, and they can then move in response to chemical signals, ionic charge, and mechanical stimulation in order to close or open. So the channels can be stuck in one place or the other, allowing things to diffuse or not. Carriers work a little different, and the whole entire protein changes shape. So they're going to have some more specific binding. That's one of the big differences. These have some specificity, but they're not highly specific. A carrier is highly specific. It has a binding site. So some molecule has to come in here and then actually bind. So it could be one, and then this thing closes and then opens up on the other side and allows this to move through from one side to the other. So it could be often a big molecule or something bigger than could ever fit directly through the fatty acid tails, or through this little ion channel, and so you can get larger molecules moving through by carriers. Again, as long as it's diffusion, it doesn't require energy, and it's also releasing energy. It's giving off energy as that happens. That's called facilitated diffusion because it's helping. The protein is just helping the diffusion process. The diffusion wants to happen on its own, and all diffusion processes, whether they're simple, directly across, or facilitated, are going to release energy because they're all where things are moving from high to low. From high to low concentration is going to be diffusion and a release of energy. Now in this particular case, the active transport, we said we're going to move the sodium, say, from low to high concentration. That will require energy. So active transporters often use something for energy. Many of them use ATP. So some of them just break the ATP into a phosphate and ADP, releasing that energy, and then the protein grabs onto it and uses it to push the other molecule. Sometimes this phosphate is physically attached to the protein and it stores energy. So the amount of energy released with ATP is 7.3 kilocalories. If the pump doesn't require 7.3, if it only requires 2 kilocalories, you have a lot left over. It's kind of like buying something and having change left over. Well, what do you do with that? Is it just lost? Is it gone, or can you reuse it again for something else? And so in this particular case, the excess, is held onto in a new bond, and then it can be broken and released a second time, so the pump can work again. Sometimes maybe in the other direction, so it can work in one direction or another. Another way, and this is going to be really important here, I'm going to explain this because I found that this is active transport, so I'm still talking about active transport, please keep that in mind, and the energy for active transport, so that's what we're getting into. There's a very important concept, right, on this exam called co-transport. And I find many people are very confused and have this wrong, and which isn't good because it's important for concepts in the third exam as well. So if you don't get it here, it really is a problem for the future in the class. If this transmembrane protein can allow sodium ions to diffuse, right, that's going to give off energy. It's released. What does that energy get used for? All kinds of things. There could be an enzyme coupled to this, and it could help a chemical reaction. There could be a variety of other things that occur. But one of the things that can happen is that the protein itself, this protein, could use that energy to move something else, I'm trying to find the right color, in the opposite direction. Right? So let's say in this particular example, there's a sugar molecule. This example doesn't make sense in the case of it's moving the sugar out and the sodium in right now, but it's just a generic example of how energy can be transferred from diffusion to active transport. Okay, so this is not a real world specific example. It would be sort of the opposite, or do it this way. Sorry, we'll do it like this. Let's say the sugar is on both sides of the membrane. You have sugars in your cytoplasm and you have sugars outside the cell. Okay? And let's say the sugar concentration inside the cell is very high, like in your cytoplasm. So this is the high concentration side, and comparatively a much larger volume outside the cell. So even though the sugar's there, concentration-wise, they're lower concentration, which means they might want to diffuse out of the cell. But you don't want that to happen. You actually want to bring these molecules into the cell for energy. So that's going to cost you. It's going to cost something to get more of these molecules, which you're going to eventually use for energy, but you're going to have to expend energy to get them. If you use the sugars to make ATP, and then you have to use ATP to make the sugars, they're not worth as much, right? So if you have to spend a penny to get $100, or you have to spend $80 to get $100, you'd rather spend the penny to get $100, a smaller amount of expenditure to get a bigger gain. Whereas if you have to expend a lot, the gain isn't very much. Okay, so in this particular case, we're using something else to get that energy, and that is going to be diffusion of an ion. Like in this example I'm giving, and there's all kinds of other examples that you could look up that are specific. Sodium ion diffuses from high to low, and then this same protein, in addition to binding a site for sodium, so it has a sodium binding site, it also has a sugar binding site. And now the protein changes shape, and as the sodium moves into the cell from high to low, the sugar moves into the cell also, but it's moving from low to high. So technically, this was active transport. Where's the ATP? There is no ATP. No ATP is involved in this type of active transport. So please, because people continually get that wrong. In this type of co-transport, it's where diffusion powers the active transport. There is no ATP directly involved in this process. So indirectly, yeah, so if there's something else that's pumping sodium out of the cell that uses ATP, and then the sodium diffuses back into the cell, technically ATP is being used at some point further down the line in some disconnected part of this system. But directly at this protein, it doesn't bind or interact with ATP. It only allows one thing to diffuse, kind of from high to low. And at the same time, it's kind of using that, as this say water level like rises up, it's kind of using that rising water level to then power something totally different. It's completely unique to move. So it's causing something else to happen. Something else is moving from low to high with this energy, whereas this is moving from low to high, that's moving. This is moving from high to low, and that energy is raising this up, and that's being transferred over here. That's just the idea to get. It's also energy coupling. So think about that, because that's a big concept on the third exam, and that's what enzymes do. They take energy from one reaction and give it to another reaction, and that's kind of what's going on here. Our link, between energetics, diffusion, and active transport. So look up co-transport, look up a bunch of specific examples in cells where they use diffusion of something to power the active transport of something else. Okay. So we've got facilitated diffusion, active transport, some different types of active transport. I'll briefly talk about bulk phase transport. Bulk phase is essentially where, instead of bringing a single ion or one sugar molecule into the cell, a cell needs to transport a large number of something in or out of the cell. So it could be either direction. And so let's say this cell here wants to bring in all these molecules. They're all the exact same molecule, and it needs a lot of them. So it can transport 1, 2, 3, 4, 5, 6, and keep transporting them into the cell across the membrane with one of these types of proteins. Or it could do something a little bit different. The cell can have on its surface proteins that are unique. They're receptors, and they bind these molecules. And so these little molecules here are now getting attached to proteins. As that's happening outside the cell, on the inside of the cell, other proteins are attaching to the other side. So transmembrane has something on one side and the other. So that means one of these things is binding on one side. I lost it. Here we go. And then something else is binding to the part of it on the other side. In this particular model, this is referred to as receptor-mediated endocytosis. So endocytosis, so cyto for cell, cytosis is movement across the cell, endocytosis is movement into the cell versus exocytosis is movement of things out of the cell. So we're bringing things into the cell. It's receptor mediated because there's a protein receptor that's controlling the process. It won't allow anything to come into the cell, and it's part of bulk phase because instead of moving one of these at a time, what's going to occur is the cell is essentially going to eat all these molecules, in a sense, by bringing them into the cytoplasm in a vesicle. So the cell is going to now change its shape and start to kind of pull itself inward. That process is directed by these cytoplasmic proteins. And then there's all these receptors, and then you have all these molecules kind of stuck in there. Eventually, what will happen is we'll have the cell, we'll of a vesicle, and inside that vesicle will be all these molecules. So all these molecules over here have now been brought into the cell, but they're trapped inside a membrane structure. And then that membrane structure can either be burst open, or it could connect to another membrane structure in the cell that has enzymes in it that break them down or transform them into something else, or whatever it is. It's totally different, unique, many different types of cellular processes can occur, but that's the idea. Bulk phase, bringing in a lot of things at once. In this particular case, it's very specific, using receptors to do it. So related to this, so I'm trying to go through the topics as fast as I can here. Remember, there's lectures on all these specifically with a lot more detail. But this is just a quick review. And it's getting long, so I'm trying to speed it up to the end. Another thing cell membrane proteins do are help cells bind together. So let's say there's two cells here, and we're not going to be moving anything across the membrane. And okay, let's say like this. So this cell has maybe this transmembrane protein, this one has this transmembrane protein. They're going to come near one another, and if those proteins recognize each other, they bind, and the cells attach to each other. If they don't recognize each other, the same as the protein binds a specific cargo molecule. Nope, it's not the right cargo molecule, it doesn't interact with it. Same thing here, the carrier. It has a specific binding site for a certain ion. Maybe it's calcium ions. A sodium ion comes in there. No, it's not going to do anything. It recognizes that it's not calcium, and so it doesn't behave. It doesn't change shape. It doesn't do anything. It just sits there and waits for something else to happen, for that molecule, the ion to go away, and something else to come in place. Okay, so if these two do not pair up or recognize one another, then nothing happens between them. But if they do recognize each other, if they are the same, that's a B. So they'll bind, right? They'll form a bond between the two cells. So this is one type of cell adhesion where we have membrane proteins binding to other membrane proteins that are like them, and they recognize each other. The particular one we talked about in this course are cadherins, which have a couple unique characteristics. They require calcium, so kind of look at the name, Ca, cadherin. If calcium is present, they're properly shaped, so they work. If there's no calcium present to interact with them, then they can't form their proper shape and folding, and so they don't function, or they don't bind. Right? So one is they require calcium. Two, they're a transmembrane protein. They go all the way across from one side to the other. And then three, think about this. The phospholipid bilayer is just an oily layer, not really held together with specific bonds, not really strong bonds. There's not covalent bonds holding these things together. They're just kind of there as a barrier. So you stick something through it and you grab it and try to yank it out. How easy is it to rip it right from the membrane? It's as easy as sticking your finger through an oily layer on the surface of water and pulling it back out again. There's nothing to really stop it from happening. So why don't these proteins just get ripped out of the membrane? They could, unless they were attached to something else. So not in this exam, but we'll cover this eventually in the class. Inside the cell is a network of protein fibers and filaments called the cytoskeleton. So cells have a skeleton made up of proteins on their interior, all different kinds. These particular receptors, that are the cadherins, they bind to that cytoskeleton. So what that means is when they bind to each other, they're not just stuck in the membrane, they're actually attached to something on the interior of the cell that, like a spider web, has many attachment points, which gives it great strength. So when you try to pull these cells together, you're pulling on this bond, and then it's being transferred through the protein to the bond between the cadherin protein and the cytoskeleton, and then it goes to the cytoskeleton to every single binding point it has that crisscrosses throughout the cell, which makes it very strong. So tissues have these types of bonds that kind of hold them together. Okay. There are other types of bonds between cells which involve things kind of like channels, but they're a little bit bigger. They're pores, right? They're openings, right, in the cell. And when these two cells share the same one or same type, they can attach to one another, and then in addition to that, form a little connection directly between the cytoplasm of one cell to the other, right? That's called a gap junction. So the gap is the pore, the opening. The junction are the binding proteins, the proteins that are holding the two cells together, right? So the cells are attached to one another, and now their cytoplasms are technically connected. So in true tissues of organisms, we have this, so the cells of a tissue can behave as one unit because their cytoplasms can be connected through gap junctions. All right, so that's kind of our cell adhesion part. Again, there's more details in other lectures specifically on that. Again, this is just a quick overview. And then finally, last topic kind of brings a lot of these things together, and that would be cell communication. So again, this is a big topic. It can be fairly complex and complicated, but what you need to get out of it is that it's going to involve the membrane structure, it's going to involve proteins in the membrane, receptor proteins, specific binding, and then sort of a relay of information. So what I'm going to do here is kind of use some of this stuff I have drawn on the board to talk about it. So in this particular case, here we have a signal molecule. A hormone, okay? Hormones that can be of a variety of different types of chemical. They could be lipid molecules like steroids, like testosterone or estrogen. They could be peptide hormones, little groups of amino acids together to carry specific signals. So biochemically, they could belong to a lot of different types of groups of molecule. The signal is outside the cell, so this is extracellular, the outside, and it binds to a receptor. That receptor then has a component on the inside of the membrane. It's a transmembrane protein. Now, there are some cases where this inner part is an enzyme, and it carries out enzymatic activity. So the signal just essentially activates the enzyme on the inside of the cell, and it carries out a job. 1, 2, and then that's it. The signal never enters the cell. And if you were to take that signal molecule and inject it instead into the cytoplasm of the cell, it does not interact with the enzyme part. It doesn't turn it on. The enzyme would stay off if that were to come into the cell. So that doesn't happen. And if it did happen, it wouldn't really trigger anything. The signal binds specifically to this receptor. The receptor then changes shape and then does something. So one of the things it can do, and what's most often, is it begins a cascade event. So many, many reactions occur, one after the other after another. And that's where this molecule here, this peripheral protein in this system, is called a G protein. And the G protein is a relay protein. It goes between a receptor and an enzyme called an effector. Okay, so we have receptor, effector. Sorry, that's really messy. I'll try to write it a little bit better. Effector. Okay. And in the middle is a G protein. So the receptor is a transmembrane protein. The G protein is a peripheral protein. The effector is typically a peripheral protein as well. So these are all embedded in a membrane, or they're attached to a membrane like this. And the signal molecule is then coming from out of the cell. It doesn't go in. And then what happens is the receptor and the G protein bind together. It's called a G protein because it then, when it's active, will pick up a molecule of GTP. So instead of ATP, for energy, it's GTP. And with the GTP molecule, the G protein can do its relay job and go to this effector, who is an enzyme. The job of that enzyme isn't to actually carry out a specific cellular activity. it's to make another messenger molecule. So that's another signal. So you have a signal outside the cell, and that's different from the signal inside the cell. This one can be referred to as a first messenger. This one, since it's inside the cell, is referred to as a second messenger. And then that second messenger signal will then bind to another protein somewhere in the cytoplasm and carry out some sort of job in the cell. So that could be binding to an ion channel somewhere in an interior structure, like an organelle, to release ions into the cytoplasm and change the charge, which is one of the examples I've given in the more detailed explanation of an IP3 pathway, which is one you should definitely focus on. But overall, communication in cells involves specific binding to membrane proteins and then the relay of information, not molecules, information into the cell. And the information is relayed through the change in shape of the proteins, not through the individual molecules. Sometimes new molecules are made inside cells that are second messengers, and then they bind to other things to carry out work or jobs within inside the cell. So, all this stuff is second exam. It's all based on the structure of a membrane. If you don't have the whole membrane structure laid out and know the difference between a peripheral and transmembrane protein, you're going to be lost with everything because it's all involving these proteins and how they work and how they function and where they're located and all of those sorts of things. So start with the structure, and then I think working in this order makes sense as well as your study. So hopefully this review it did go much longer than I planned, but hopefully it helps you as you study.